amazing. Okay, so it's 12, and I know we're all probably starting to think about food a bit. I know I am. But before we move on to lunch, and we also have some amazing behind-the-scene tours for you, we've actually got a series, Flash Talks, for you, which is going to be exciting and amazing. So our first Flash Talk of the day is the president of the Horticultural Society at Edge Hill, our very own Felipe Galtarossa.
So sorry if I butchered your surname there. Which one is it, babe? It should be. It should be that one. Is it this one? I think. Oh, yeah. Yes, that looks like it. Yeah, it is. Yeah, right. Can I just zoom in and back? Are you happy with that? Is it? Yeah. Yeah, beautiful. Right. There you go. Do you want me to clip it on for you? Yeah. Okay.
So, you've got a minute.
Yep.
And I'll get closer to you as the minute decreases. Okay. Take it away.
Hi everyone, my name is Felipe Galtarossa. I'm the president of the Horticultural Society.
Try now.
Is it on?
Yeah. Can you hear me now?
Yeah. Great. Well, yeah, I'll introduce myself again. Hi, my name is Felipe Galtarossa. I'm the president of the Horticultural Society. And I'm up here because I wanted to share more about what we do and who we are. So, yeah, we're a student-led society based in Edge Hill University that creates opportunities for students to get involved in green spaces, discussions in plant-related topics, and hopefully create a wonderful, inclusive community to share our love for botany and horticultural and plant-related topics. And our goal for the society is just to create a wonderful network. Our goal for today was to create a wonderful network of plant societies to share resources, practices, knowledge, ideas, and our love of plants. I think we've put our email in socials, so come find us online. I think that's it.
Fantastic, thank you very much. Oh, that was great. Okay, so our next talk is Giorgia Irlam from Edge Hill University. And this research has a particular place in my heart because we've been working together on it and I love it. So, which one is yours, sweetheart? I'm going to guess it's someone with your name. Come on, and then I'll zoom in for you. Okay, so let me give you it. Just hold that. And I'll put this on you.
I don't really have that much to say. So.
Well, it's a flash talk, mate, so you're doing great. Take it away.
Okay. So my research, I've been doing it here at Edge Hill, is on Sorbus lancastriensis. Sorbus is a diverse genus of plants, and we have 43 native Sorbus species in the UK and 35 are endemic. So Sorbus lancastriensis is endemic to Morecambe Bay, and there is also a population in North Wales. I've been looking at genetic diversity within and between populations of the Lake District populations. I've got a poster outside. Everyone can come and talk to me if you'd like. That's it. I don't really know what else to say. I'm sorry.
It's flicking away from me. She's got that.
Oh, you did amazingly. Oh, yeah, that would be great. Thank you very much. Amazing.
So, our next talk is... Oh, sorry. Like this one. Oh. So our next one is Ella Chesterton, also from Edge Hill University. Let's have a quick look. I think it's this. Yes, excellent. So here you go, Ella. You take it away, my girl.
Thank you. So my poster is on my current
Ella?
Good? Better, nice. Okay, so my poster is on my current MRes project that I'm working on this year. It's on Carex species, and I'm looking at whether their form is more affected by their phylogeny, so their ancestral characteristics, or by their function, so whether they're more adapted to their habitat. I've also got some herbarium ID that you can come and have a go at, and matching that to the leaf section. Some more of my sketches, if you'd like to see them. You can find me at exhibit number 7, so come and say hi.
Fantastic. Thank you very much, Ella. Okay, so our next one is the lovely Maddie Fyers from Edgehill University.
Oh, first go. Oh, here we go. Okay, right.
Take it away, Maddie.
Hi there, my name is Madeline Fyers and I'm an MRes student here at Edgehill, and I'm studying the population genetics and connectivity of the rare aquatic plant Luronium natans. So what makes this plant, or what contributes to this plant being quite rare, is the fact that it's got quite a variable morphology. I've put 2 examples of how varying this morphology can be on the board behind me. And that's one thing that factors into it being quite rare. Another thing is the fact that the populations are really quite isolated, which is sort of weird for an aquatic plant. So I'm studying how these populations relate to each other using a genetic region called microsatellites. And that will give me an idea of where each individual comes from across the UK, and hopefully where the centre of diversity is for this species. But if you'd like to hear more about it, come and find me at my poster outside. I'm at exhibit 20. Yeah, thank you so much.
Thank you very much, Maddie. That's fantastic. Okay, so our next talk is the lovely Jennifer Wilkinson.
Thank you, darling. Hi, everyone. I've just completed my MSc here at Edgehill, and I've been studying Limonium britannicum subspecies celticum, the Celtic sea lavender. It's a gorgeous and rare little halophyte on the northwest and North Wales coast. It began as a bit of a sort of treasure hunt, going back 25 years in the records, finding where it was and finding it to be present in 9 sites. And what I did was the first sort of range-wide population survey, and some really interesting stuff came up. The key one is that it recruits, so new plants are much more frequent on sandy soils than they are on rock. And that's important because if we know where it likes to regenerate, then we can protect it, remembering that our coastal sites are under increasing pressure and they're changing fast. So if you'd like to know more about Celtic sea lavender, I love talking about it, so please come and visit me at Exhibit 33.
Oh, no, that was fantastic. Thank you. Oh, so let's keep the ball rolling. And so next we have Kaitlin Saward. Am I saying that correctly? I'm butchering names today. I do apologise.
So, darling, which one is KS? Ah, thank you. Fabulous, thank you. Thank you very much.
So hi, I study hybridisation, and my hypotheses all relate to a potential relationship between genetic divergence and hybridisation. And to study that, I'm making loads of crosses of epilobium, and you can see one here. So I've tried crossing lanceolatum and rosium, and you can see some intermediate morphology just from the flower, where the stigma shape is a bit sort of in the middle and a bit messed up. And then I'm looking at measures of fertility in those, and as you can see for this cross, the seed set, the pollen fertility and the germination percentage is all lower in the F1 hybrid, and then the seed set is even lower again in the F2. And my eventual plan is to be comparing that between loads of parental pairs and ones of high divergence and ones of low divergence. Yeah, so if you want to hear any more, my poster's at exhibit 19. Thank you.
thank you very much. That was fantastic. Okay, and next up we've got Wilson Flint, another one of Edge Hill University's own.
I was also stupid enough to not put my name on file, so...
It's your students, Paul. To have a word.
Right, how do you...
Oh, beautiful. Just press zoom in. Just zoom in. Anyway. You happy?
Yeah,
Beautiful. All for you, darling.
Hello, everybody. So I am another master's student at Edge Hill. I'm doing my master's project on limestone grasslands in the Lake District and hopefully the Yorkshire Dales soon next year. So limestone grasslands are one of the most diverse habitats in Europe, and in fact on a small scale, under 100 metres squared, they can actually outbeat tropical rainforests, so really quite diverse areas here. So obviously that means, I don't need to explain to you why that means we should protect them. So my project is looking at the different management styles to try and increase species richness and diversity across limestone grasslands. So I've got 3 treatments. I'm looking at grazed ones, ungrazed ones, and grasslands in which grazing's been reintroduced, just to try and see at what point grazing will increase species diversity. So I'm recording all species, their percentage cover. I did start doing sward height as well to try and get an understanding of the grassland itself. So hopefully I'll have some exciting data next year. I'm at exhibit number 23. If you want to come and say hi, come and say hi.
Thank you. That was a bang on a minute. Well done, mate. And then next we have the lovely Ivy Harris, also from Edge Hill University. Which one is it, babe? We've got this top one. Should we go with that one? We'll try that one. And if it's not the right one, is that it? Yeah, beautiful.
All right. Hi everyone, I'm Ivy Harris from Edge Hill doing an MRes this year. I'm looking at Project Glade, the grassland aquatic drought experiment. I'm looking specifically at microbial community diversity on the phyllosphere, which is the surface of the leaf and anything above the ground. I'm looking specifically at Bacillus thuringiensis cry genes, which is a type of pesticide, and I'm looking at how they differ between drought. So basically what I want to try and do is anticipate like a computational model of what will happen in drought with these different strains. If you've taken a walk, or if you do, you'll probably see the huge experiment. So yeah, come find me at Exhibit 22.
Amazing. Thank you very much, Ivy. Okay, and then next on our list is Dr. Trevor Dines. Okay, let's have a look. And oh, there you go, sweetheart.
I seem to be here to prove that old people can do a flash talk.
Do you
Maybe
have
not.
a page, sweetheart?
Yeah, Trevor Dines, BSBI Trevor Dines, flash talk. That one.
Fabulous.
Yeah. Shoot me down if I'm over a minute.
There you go, happy?
So I've been rather obsessed with Welsh Groundsel over the last couple of years, a plant that Paul Ashton knows very well. And I think because it's a weed, it's been rather overlooked in the last couple of years. It's an endemic species, unique now to Wales, found nowhere else in the world. And populations we've sort of taken our eye off the ball. Populations have crashed. This year, there were just 52 plants. That's not enough for every botanical student in Britain. So it really is a rare species. We're very good at driving it to extinction because of our obsession with neat and tidy. So we're great at herbiciding it. We're great at strimming it. We're great at developing industrial parks where it used to grow. I also think nitrogen deposition from cars is having a big impact. But the most heartbreaking moment in my botanical conservation career was outside Chirk Town Council Hall, where the week before I visited, the caretaker had been along the hedge where there were the largest plants in the world, picked about 20 of them and discarded them on top of the hedge. That's how to get rid of a species that's endemic to Britain. So with the Species Recovery Trust and Natural Resources Wales, we're now launching a plan to bring this beautiful thing back from extinction. Thank you.
Thank you very, very much. Oh, fantastic stuff. Okay, and our next speaker is the lovely Rachel Murphy from a National Plant Monitoring Scheme.
you at the bottom. There we go. Amazing. Is that it? Yeah, that's it. Perfect, thank you. Hi everybody. So yes, I'm Rachel Murphy. I'm with the National Plant Monitoring Scheme, which is a nationwide citizen science project with hundreds of volunteers heading out twice a year, every year, to survey their allocated sites for indicated plant species. We're actually celebrating our 10-year anniversary this year. I was hoping for a whoop, but that's fine, we'll go without it. And we've been celebrating some of our amazing achievements over that last decade, from our growing data set of over 250,000 species records, over our 8,000 plots, our expanding and evolving training programme, and of course our amazing community of volunteers without who it would not be possible. We are contributing MPMS data to the UK biodiversity indicators, along with over 600 other studies to date. So it's an incredibly impressive data set that just would not be possible without the volunteers. If you are interested to find out if there is an MPMS survey square near you, feeling a little bit lonely, looking for somebody keen to go and visit it twice a year and survey, do take a look at our squares map on the website, or even better, come and chat to me at Exhibit 40 and we'll have a look to see if there's a square near you. Thank you.
Oh, awesome. Thank you very, very much. And finally, but by no means least, we have the lovely Kieran Heggs from Edge Hill University. Kieran, along with Kit, has done so much work for this conference, so thank you so much for all the work you've done just in advance.
I'm fairly sure mine doesn't have my name on it either.
Are you Paul's student as well?
No, I'm Faye's.
I'll have words with Faye. Which is it, babe?
That's probably one of these.
Who do you think? Should we go that one? Yeah. First go. Oh, we're doing well at this. Take it away, my love.
Hello, I'm Kieran. I am a third-year plant science student here at Edge Hill. I am studying the mycorrhizal and Dryopteris dilatata broad buckler fern for my dissertation. I have a poster outside, but I'm running around here, there, and everywhere today. So if you do have any questions and you can't catch me, make sure you send me an email. Thank you.
Thank you very much, Kieran. Okay, fabulous. So, with that, thank you so much to all of this morning's speakers. It's greatly appreciated you all coming up and sharing your knowledge with us. We're now going to be breaking for lunch. Yay! So, you'll need to turn left when you come out of the lecture. Walk along the hallway until you come to the stairs. There's a lift on your left if you need the lift. Tea, coffee, sandwiches and biscuits have been laid out for you. And if you are joining one of our behind-the-scene tours, which is the SEM suite with the amazing Lorna and the fernery with the lovely Kieran, you'll need to assemble at the BSBI stand near the entrance. And we'll see you all back in here for 1:30 for our second round of talks. Maybe just hold on for a minute. Do you need to say something?
Can I?
Yes, of course.
If anyone who has been involved in Botanical University Challenge in any way as a student, speaker, workshop leader could stay behind for 2 seconds, we'd love to get a big group photo.
Yes, amazing. Amazing. Anyone else got any more announcements or? No? Fabulous. Thank you very much, guys. We'll see you after lunch.